Dear students, welcome back to our official YouTube channel SR and BGNR Government Arts and Science College, Khammam. I am Dr. B. Satish Kumar, Assistant Professor in Biochemistry. From last few video lessons, we have learned about the replication process in molecular biology, that is replication process and transcription, post-transcriptional modifications. So these three topics we have covered so far in the unit 1 of paper 7, final year students, semester 6. Okay, so in the second unit we have covered the genetic code and translation and post translational modifications. So now we came to the unit 3 of molecular biology paper, which is regulation of gene expression. So in this video lesson we are going to cover some of the topics from this unit, that is regulation of gene expression. In the regulation of gene expression, the syllabus that we have is regulation of prokaryotic gene expression. So there are two parts in prokaryotic gene expression regulation, that is induction and repression. So we will go into details and we will learn what is induction and what is repression process with specific examples. Okay, and the second subtopic is lactose operon and catabolite repression. Third one is tryptophan operon and attenuation. So fourth topic is positive and negative regulation. So when we look at the entire structure of this unit, we are going to cover only 4 basic things. First one is lactose operon, second one is tryptophan operon. So lactose operon comes under the example of induction, and tryptophan, it is example for repression process. Okay, these two operons, very important operons. So along with these two operons we have some more further regulatory processes in the lactose operon and tryptophan operon. So for lactose operon we have catabolite repression, and for tryptophan operon we have transcription attenuation. So only 4 topics are there. Don't be confused with all the terminology that is given in the syllabus. So only 4 topics we are going to cover. First one is lactose operon and catabolite repression. Second one is tryptophan operon and attenuation. Okay, so the first topic is nothing but just an introduction, and the induction and repression itself. The lactose operon, tryptophan operon are the examples for induction and repression. Positive and negative regulation also the same examples will be given, but that is just a general discussion. Okay, positive and negative regulation. So we will have the same examples for positive and negative regulation also. So don't be confused with this first topic and fourth subtopic. Only the topics are the 3.2 and 3.3. These are the main topics only. Okay, so now let us start with introduction to gene regulation of gene expression. Okay, so first of all we will start with what is gene expression. Okay, gene expression is the synthesis of gene product. What is the gene product? What is the ultimate goal of a gene? So gene is nothing but a segment of DNA which encodes a polypeptide chain, or which has information for the synthesis of a particular polypeptide chain. So it will have sequence of nucleotides. That sequence of nucleotides corresponds to sequence of amino acids. Okay, that is nothing but a polypeptide chain. So each and every gene contains a sequence of nucleotides. That nucleotide sequence specify a polypeptide chain. So that is utilized for the synthesis of that particular polypeptide chain or amino acid sequence. Okay, so every gene uh, in one way or other way, so the genes are nothing but they are the information for the synthesis of polypeptide chains. Okay, so but we, as we all know, the genes directly do not involve in the synthesis of polypeptide chain. So directly it will not enter into the translation process. Okay, so the genes means DNA. Okay, so that DNA, the information at the DNA level doesn't translate into polypeptide chain directly. First of all, a copy of the gene is prepared in the form of an mRNA, and then only that mRNA enters the translation process and a polypeptide chain will be synthesized based on that mRNA molecule. Okay, so that is why we have two processes in the gene expression. So first one is first first we have the DNA. DNA is nothing but gene. In case here it is gene. Okay, this is particular gene, and this gene is specific for a polypeptide chain or a protein. That name is, suppose it is X protein. So now this gene is X gene, and this gene enters a transcription process. Okay, transcription is nothing but DNA dependent RNA synthesis. So from this gene we will get an mRNA molecule. So we will have same nucleotide sequence in mRNA and DNA also. Okay, same sequence, but only difference is DNA contains uh, T, thymidine, and in case of RNA we have uracilin instead of that. Okay, only that difference is there, and mRNA in fact that is single stranded and DNA is double stranded. Okay, some uh, basic differences are there, but anyway it copied the sequence of nucleotide from the gene is copied into RNA and mRNA molecule. So that is what transcription. So after transcription, that mRNA enters translation process. Okay, translation is nothing but translating the nucleotide sequence into amino acid sequence. Okay, this is what translation. So in the previous videos we have learned about the entire process of translation, where we have four different stages, and first stage is activation of amino acid, second stage is chain initiation, then chain elongation, chain termination. So all these stages comes under translation process, in which from mRNA a particular polypeptide chain will be synthesized. Okay, so that is translation. So now what is gene expression? So gene expression means how the information at the GNA level is translated into the amino acid chain. Okay, so that is nothing but gene expression is transcription plus translation. Okay, transcription plus translation is called gene expression. From the gene we are getting the product, that is a polypeptide chain. So that is gene expression. So this one is okay. So if we take for example, if we take human being, that human being, each and every cell of human being contains around 50,000 genes. Okay, each and every human being, the cell of each and every cell of a human being contains around 50,000 genes. Okay, when we look at all these 50,000 genes at any particular moment, only a few genes will be in active form. Only few genes will be expressed and remaining all genes will be repressed. That means they are in switched off form. Only a fraction of genes will be in switched on form, and remaining all the genes will be in switched off form. So how this switched on and switched off going on? Why some genes, how some genes are being switched off, and why some genes are how they are switched on? So that is what the regulation. So whenever a specific enzyme or a protein is required, then only that particular gene is expressed. That means that gene is switched on and that expressed to produce the needed, the necessary polypeptide chain only. Okay, otherwise it won't start synthesizing, uh, or it won't express to form that unuseful unusable synthesis, because that will waste, that will cost uh, the energy of the cell. Okay, if you don't want uh, one particular gene product or a protein, then you should not synthesize that. Otherwise you will lose much of the energy in synthesizing wasteful or not useful products. Okay, so this is for the cellular economy. So for the economy of the cells, they will they will switch on and switch off genes based on their need only. Okay, so and that is why it is called regulation. Regulation is nothing but when when uh, when should the genes must be switched on and when should they must be switched off. So that is called the gene regulation. Okay, so the gene regulation, where does the gene regulation occurs, whether that occurs at a transcription level or translation level. So because we have in gene expression we have two processes, transcription and translation. But here in case of prokaryotes, the expression, yeah, of course in case of eukaryotes also, the regulation of gene expression occurs mostly at the level of transcription. Okay, so at the level of transcription, if the transcription is switched off, then what happens? Translation is also will be switched off because there is no mRNA product. So, okay, so that is why at the first stage itself it should be regulated. So most of the regulation process takes place at a transcription level. So, that transcription level, uh, there will be some signals. Okay, there will be some signals which recognize the necessities of the cell, and they will either switch off or switch on the genes, and they are called transcription factors. Some of the transcription factors will regulate the expression of genes. So they are under the influence of signals from the environment. Okay, suppose the best example that we are going to discuss anyway in this video lesson is lactose operon. So if you take the lactose operon of E. coli cell, if you take the E. coli cell and if we grow that E. coli cell on a agar medium, and if we add lactose as energy source for that bacteria, then what happens? That should take up that lactose and that should metabolize that lactose to produce energy. Okay, otherwise if there is no lactose, then it should not uh, mm, produce any enzymes, the enzymes which involve in the lactose metabolism. So whenever the lactose is there in the medium, then only the lactose utilizing enzymes must be synthesized, otherwise they must be repressed, or they must the genes for those enzymes must be repressed. Okay, so here that signal is coming from the surrounding environment. So we are adding externally, we are adding lactose to the medium. Then what happens? That is sensing, the E. coli organism is sensing the presence of lactose, and then it is switching on the genes, and that genes are producing, expressing to produce specific metabolic enzymes, and those enzymes mm, will take up the lactose and hydrolyze or metabolize for the production of energy. Okay, so that is why the regulation of gene expression is monitored by or is controlled by the signals from the environment. Okay, so that signals are conveyed to the specific genes via transcription factors. Okay, and in regulation of gene expression, the well studied example comes from Escherichia coli, that is E. coli. It is prokaryote and bacterium, and that bacterium, in that bacterium we have studied lactose operon, lactose operon in detail. So that led the scientists to find out or to know about regulation of gene expression. Okay, so first of all, uh, when we look at the genome of Escherichia coli, we will find out two types of genes based on their expression status. Okay, so some genes are called constitutive genes. Constitutive means continuous. So they are also known as housekeeping genes. They must be produced continuously. They are required every second. Okay, continuously. As long as the bacterium is live, these constitutive gene products are very, very necessary. So therefore, they constitutively means continuously express these genes and produce their corresponding proteins, which are essential for the maintenance and life of E. coli cells. Okay, so that means there is no regulation for the constitutive genes. They don't depend on the environmental factors. Okay, so they will be continuously produced, but the remaining proteins, sorry, remaining genes, which are called regulatory genes, so regulated genes, so these regulated genes will synthesize some of the proteins only at a particular condition, particular situation only. Okay, so that means they are regulated by the environmental cues. Okay, so they are called regulated genes. So the enzymes which are produced by the regulated genes are called regulated enzymes. Okay, so uh, they are only necessary during a particular time based on the environmental condition. They never be useful constitutively or continuously. So that is why, so whenever the need arises, then only those enzymes are synthesized by gene expression. Okay, so here regulation of gene expression is divided into two types based on their mechanism. So first one is induction and second one is repression. So the name itself, we can easily understand what is induction. Induction means induce. So something is inducing the expression of genes. Okay, something is turning on the genes, so that is called induction. So that means that those genes are already in turned off state, and so something is turning on those genes. So that is the phenomenon of induction. And what about the repression? So here these genes are already in switched on form, but something is turning off those genes. So this is the difference between induction and repression. So in the induction, the genes will always be in switched off form. So only in the presence of the specific signal here, this the example for this is lactose operon, and presence of lactose acts as the inducer. Okay, so lactose operon will always be in switched off form. Only in the presence of lactose, that lactose will switch on the genes. Okay, so that is why lactose is inducing the genes, switching on the genes. So that is why it is induction. And in case of repression, we have the example of tryptophan operon. Tryptophan is it is an amino acid. So it is required for the production of polypeptide chains. Okay, so this is a biosynthetic um, operon. Okay, so uh, these kind the amino acids are uh, necessary. The amino acids must be provided, enough amino acids must be provided to the cells for their continuous production of different kinds of proteins. Okay, in any given time, um, mm, a few or more polypeptide chains will be continuously synthesized in cell. So whatever the polypeptide chain or whatever protein it may be, it requires 20 different types of amino acids. So those 20 different amino acids must be synthesized continuously. Whenever the uh, production of a particular amino acid goes beyond a limit, then only the production must be slowed or it should be stopped. Okay, so that is why in repression, the example is tryptophan operon. The tryptophan operon is always in switched on form. It continuously synthesizes the tryptophan because it will be continuously utilized. Okay, consumption also takes place continuously and production also takes place continuously. Suppose if the production rate is more and consumption rate is less, then what happens? The the standing state of the tryptophan, okay, the standing state of the tryptophan goes beyond a limit. Okay, so now that genes must be switched off, otherwise it will waste the cellular energy. Okay, because that is not no longer necessary up to some time. Okay, so that is why it should be switched off. So the tryptophan operon is always in switched on form, but when the tryptophan levels reaches or goes beyond the limit, so then that tryptophan will stop the expression of genes. So that means the tryptophan will switch off the genes. That is nothing but repression. So here the tryptophan itself acting as a repressor, and in case of induction, lactose is itself acting as the inducer. Okay, so let us start with the induction. Okay, so now uh, in the previous slide we have just looked at what is induction process. Okay, so let us quickly see that when lactose is present in the medium, so that means suppose you have a test tube. Okay, in this test tube you have growth medium, and in the growth medium you have several E. coli cells in it, several E. coli cells. And what happens, they utilize the growth medium provides all the essential nutrients for their maintenance and for their growth, for their growth. So what happens, for every 20 minutes, for every 20 minutes the number of cells inside this tube becomes double. For every 20 minutes it becomes double, because the life cycle uh, of E. coli cell is around 20 minutes. For every 20 minutes it divide and form two cells from a parent cell. Okay, so continuously it grows. So for this we have to add all the essential nutrients like carbohydrates and proteins. Okay, so and some minerals, and we have to adjust the pH, and so everything. So here when we add lactose, okay, lactose, when we add lactose as energy source or carbohydrate source, then what happens, this E. coli must utilize this lactose, and this lactose must be metabolized metabolized to form energy. Okay, to produce the energy. Okay, for so it acting as a energy source. Lactose is acting as an energy source, energy and material source. Okay, it is just food material. Now it is food material for this E. coli cells. Okay, so when we add lactose to the medium, then what happens, it start using the lactose, and the bacteria will grow. Okay, when we when the lactose is finished or depleted in the medium, then what happens, it won't synthesize any enzymes which are essential for the metabolism of lactose. Okay, so it will shift its attention towards other source. So if we add, after depleting the lactose, if we add glucose to that same medium, then what happens, it will stop the synthesis of lactose genes and start synthesis of the genes which can metabolize glucose, because now glucose is present, not the lactose. Okay, if lactose is there, only lactose is there, then it start using the lactose. When the glucose is there, then it stop utilizing, stop the lactose genes and start the switch on the genes responsible for glucose metabolism. So this is what the induction. So when we add the inducer, that is lactose, to the medium, that induces expression of genes which metabolize the lactose. Okay, so therefore here the substrate itself acts as an inducer. Okay, so here when you look at the general concept of lactose operon, okay, when we add lactose to the medium, then what happens, the lactose operon, so there is a operon known as lactose operon in E. coli. Operon is nothing but it contains a set of genes, set of regulated genes, and that set of genes are essential for a particular metabolism. So we have lactose operon. That lactose operon contains three genes: one, two, three, Z, Y, A. So these three genes are there, and these three genes produces three proteins, and these three proteins are essential for the metabolism of lactose. Okay, so that means these three genes are not scattered throughout the genome, they are at one place. So they are uh, like a single set. Okay, just adjacent to one another. They are like a continuous sequence, they are not separate genes. Okay, only one long nucleotide sequence. From that you will get three gene products Z, Y, and A. Okay, so these three genes are essential for the metabolism of lactose, and this these three genes and along with their regulatory sequences are known as a operon. So in the lactose operon, we will have three structural genes. So here, what happens when we add lactose to the medium, that lactose must enter into the cell. Okay, for that lactose need a permease protein, a permease protein that allows the entry of only lactose into the cell. Otherwise the cell won't take up the lactose, because lactose is a big size, big molecule, large molecule, and that is also a polar molecule, and that kind of molecules are not directly passed through the cell membrane. Okay, it requires some assistance like permeases. So we have galactoside permease here, galactoside permease that allows the entry of lactose into the cell. So once lactose enters into the cell, the lactose actually it is a disaccharide which contains beta D glucose and beta D galactose, beta D glucose and beta D galactose. So therefore this must be the glycosidic bond, beta glycosidic bond between the glucose and galactose must be broken. Okay, so that is why that lactose is hydrolyzed to form glucose and galactose. Okay, glucose and galactose. So the enzyme which involved in this breakdown is the beta galactosidase. Okay, so here for the lactose metabolism we require two important proteins. First one is lactose permease or galactoside permease that allows the entry of lactose into the cell, and the second one is beta galactosidase that breaks the glycosidic bond between glucose and galactose in the lactose to form glucose and galactose. Okay, so along these two are important. So that is why when the lactose gene, lactose operon is active, then these two genes are expressed to produce these two proteins. Okay, namely galactoside permease and beta galactosidase. So these are the essential genes here. Okay, so that is about the E. coli cell lactose metabolism. Okay, when we look at the structure of lactose operon, as we have already discussed that uh, lactose operon contains three structural genes. Okay, three structural genes. First one is lac Z gene, this one lac, then lac Y gene and lac A gene. Okay, so this lac Z gene is the important gene that produces beta galactosidase. So what is the function of beta galactosidase? Beta galactosidase will hydrolyze the lactose into glucose and galactose. So this is the key enzyme that involves in the metabolism of glucose, lactose. And the second one is galactoside permease. So this, the function of this protein is it allows, it forms a channel through the membrane, cell membrane, through which the lactose enters the cell. Okay, otherwise if you don't have the permease molecules, so then what happens, even though the lactose is present in the medium, that won't enter into the cell. If that lactose doesn't enter the cell, then the beta galactosidase inside the cell cannot metabolize it. Okay, first of all it should enter into the cell, then only the beta galactosidase cleaves or hydrolyze the glycosidic bond to form the monosaccharides, that is glucose and galactose. Okay, so permease that allows the entry of lactose into the cell. And one more is there, that is lac A gene that produces one enzyme that is trans-acetylase. Okay, we don't know what is the exact role of the trans-acetylase, but it was believed that trans-acetylase, it is an enzyme which detoxify some of the unwanted material that enter through the permease. Okay, so that bacteria is living in an environment where some of the toxic elements, suppose if there are some toxic elements, those toxic elements may also enter into the cell through the permease molecule, permease protein. Okay, sometimes it may happen. So if those uh, toxic materials are not detoxified, then what happens, that will harm the bacteria. So to detoxify the unwanted material which entered into the cell through the permease must be detoxified. Okay, that detoxification requires one enzyme. That enzyme is trans-acetylase. So these are the roles of three structural genes. So now we have the structural genes, uh, and these structural genes, okay, in the presence of lactose, these three structural genes must be expressed. Okay, so now they should be regulated. That means there should be some switching on and switching off mechanism for the structural genes. So that is why we have a promoter site. We have a another gene which acts as a switch to the structural genes. Okay, so that gene is lac-I gene. Okay, lac-I gene, that is nothing but a repressor gene. Sorry, that is a repressor gene. So lac-I gene that produces what? A protein. That protein is called a repressor protein. Repressor means that will switch off. Okay, that stop, repress. Okay, what does it stop, we are going to see later. So a repressor protein. So now when the lac-I gene is active, that expresses to produce a repressor protein. So now we have a repressor protein here. Okay, the role of the repressor protein we will discuss now. And so this is the regulatory gene. So along with the regulatory gene, we have promoter sequence and operator sequence also. Okay, so what is the promoter sequence? We already know that we have seen about the promoter in transcription. Okay, in transcription we have discussed about the promoter region. Promoter is the region where the RNA polymerase binds. So we have -10 region, -35 region. So all these things we have already discussed in transcription process. So here there must be a promoter site for each and every gene, otherwise transcription won't take place. Okay, so promoter is compulsory. So along with the promoter we have another sequence known as operator. Okay, so promoter, now it is written as P lac. So lactose promoter it is. Then we have operator sequence. Okay, operator sequence. So operator sequence is the actual key, actual switch. Okay, this switch is under the control of this repressor protein. Okay, so that is the switch. So now what happens when the repressor is synthesized? This repressor is active repressor is synthesized from the lac-I gene. Okay, so now this repressor, what happens, it comes and binds to the operator. Okay, so operator sequence is present inside the promoter sequence, in fact. Okay, so the promoter sequence and operator sequence both are not two different sequences which are placed a little bit apart, but in fact it is the operator itself a small segment of promoter sequence. So that is why when this repressor comes and binds to the operator, then what happens? Now the promoter site is occupied by the repressor. So therefore the RNA polymerase cannot bind to the promoter site because it is occupied by the repressor protein. So now that repressor is inhibiting or physically stopping the entry of RNA polymerase into that region, promoter region. Okay, so that is why it is the operator sequence. So if the repressor is there, then it stops, it binds to the operator and stop the entry of RNA polymerase, and ultimately it stops the transcription. Okay, if repressor is inactive, then operator is free, then RNA polymerase binds to promoter site, then it undergoes transcription, and finally the product will be formed. So this is the basic structure of the lactose operon. So in this case, lactose operon, we have two important regions. Okay, first one is the structural genes. In that structural genes, we have three genes: LAC-Z gene, LAC-Y gene, and LAC-A gene. Z for beta galactosidase, that metabolize lactose into glucose and galactose, and LAC-Y genes that produces galactoside permease, which allows the entry of lactose into the cell, and LAC-A gene that produces transestylase, which detoxifies unwanted material entered along with the lactose through the permease. Okay, so that is about the structural genes and the regulatory sequence, regulatory gene and regulatory sequence. So regulatory gene is LAC-I gene that produces a active repressor molecule that binds to the operator. Okay, the function of the repressor is binding to the operator. Okay, and the regulator sequence are promoter and operator. So promoter, the role of the promoter is just that binds the RNA polymerase and start the transcription, and operator, role of the operator is it binds the repressor molecule. So that information is given in this slide. So this is the lac so here the LAC-Z gene, LAC-Y gene, LAC-EA gene, and control region. In the control region, we have LAC-I gene and promoter and operator sequence. So all we have discussed in the previous slide itself. And let us start how this lactose operon is regulated. Okay, so we have two situations. Okay, first one is absence of lactose. What happens in the absence of lactose? How the structural genes are switched off. Okay, and the second situation is how the presence of lactose will switch on the genes. Okay, so now we will see in the absence of lactose. In the absence of lactose, what happens? A active repressor, the LAC-I gene, is expressed. First of all, it undergoes transcription and produces an mRNA, and it produces a repressor protein. So this repressor protein is active. It is active. So active repressor is produced. That active repressor binds to the operator site. Okay, so because of that repressor at the operator site, the RNA polymerase cannot enter into the promoter site. So if there is no RNA polymerase, then there is no transcription of structural genes, and ultimately no translation and no proteins will be produced, because we don't need the structural gene products because the lactose is absent. Okay, so now second situation is presence of lactose. So now what happens? Now the lactose is there, and we have the active repressor. Now that lactose comes and binds to the repressor. Okay, and inactivate it. Okay, so lactose, it is an inducer that comes and binds to the repressor molecule and make it inactive. Because of this inactive complex, that is inducer and repressor complex, which is inactive repressor, that is unable to bind to the operator site. So now the promoter site is free. RNA polymerase enters the promoter site and it transcribes the structural genes to produce a mRNA, a long mRNA which contains three structural genes. Okay, this kind of mRNA is called polycistronic mRNA. Okay, from this mRNA, three gene products will be produced. First one is beta galactosidase, second one is permease, and third one is trans-acetylase. So, polycistronic mRNA, from the polycistronic mRNA, we will get these three polypeptide chains, or ultimately the proteins necessary for the metabolism of lactose. So this is what happened. So there is one more important point is that allolactose is there. What does it means, allolactose? So allolactose means uh, when in the absence of lactose, the beta galactoside, the amount of beta galactosidase, the enzyme quantity of beta galactosidase will be very less because there is no lactose. Okay, but still there exists some amount of beta galactosidase. So that is why the concentration of beta galactosidase is very less in the absence of lactose. Once we add the lactose to the medium, then what happens? Lactose slowly enters into the cells because the number of permease molecules also very less. Okay, slowly it enters into the cell. Once it enters the cell, because of less concentration of beta galactosidase, the lactose is not converted to glucose and galactose directly. So first of all, it converted to allolactose. Okay, so the activity will be less, therefore the lactose is converted to allolactose. So that allolactose acts as a strong inducer more than the lactose. Okay, so that allolactose comes and binds to the repressor molecule here. Okay, so that lactose is converted to allolactose and that allolactose binds to the repressor, therefore it becomes inactive and the transcription genes are switched on and transcription translation takes place. So now we will get a lot of amount of beta galactosidase and a lot of amount of permease. So then what happens? This increased amount of permease concentration allows more lactose into the cell, entry of lactose into the cell, and high concentration of beta galactosidase that break the lactose into glucose and galactose. So that means when we initially add the lactose to the medium, then what happens? That lactose slowly enter the cell and that lactose is converted to allolactose by the beta galactosidase because that is in less concentration. Okay, so that allolactose is the actual inducer that inactivate the uh, repressor molecule. Okay, so this is what the functioning of lactose operon. Okay, let us go back to this slide. So here this beta galactosidase at lower concentration it converts the lactose into allolactose. So this is the allolactose. At a higher concentration it converts the lactose into galactose and glucose. Okay, so this allolactose and the lactose itself, if these both act as inducers that bind repressors and make that repressor inactive. Okay, this way the regulation of switching on switching off mechanisms takes place for the lactose operon. So simply once again I will repeat the switching on and switching off mechanism. So in the absence of lactose, repressor is active and that repressor binds to the operator and stop the entry of RNA polymerase into the promoter site. That turn off the structural genes and no gene products will be formed. And in the presence of lactose, the lactose may be converted to allolactose and the lactose along with allolactose acts as inducer that binds repressor and make that repressor inactive. So that inactive repressor is unable to bind the operator site and now the promoter site is free. That RNA polymerase enters the promoter site and transcribe the structural genes. After the transcription, again it takes that polycistronic mRNA enters the translation process and produce the corresponding gene products, that is beta galactosidase, permease and transestylase. Beta galactosidase that hydrolyze lactose into glucose and galactose, permease that allows the entry of lactose into the cell, transestylase that detoxifies unwanted materials that entered into the cell along with the lactose through the permease. So this is what happens during the regulation of lac operon. Okay, thank you all.